Hello, this is Miss Dagenford, and I'd like to switch our attention to the nervous system, and that still falls under regulation, but now we're going to look at how electrical signals are sent to regulate the body. Now, before we do that, we need to establish the basic parts of a neuron. And so these are all the major parts that you need to understand to know what's going on. So, for instance, if we have our structure of our cells up here, we have our dendrites, and the dendrites are going to be these long branch-like structures that come off of the cell body. Now, for some of you, this might be memories rushing back from last year. Here's our cell body. This is the main portion of the cell. We can have a quick review. Here's our nucleus and the rough ER and all of the major parts of the cell that we normally see. Now, the axon is going to be this very long structure right here, and the axon can be for instance, in the human body, it could be the length of one neuron, and that might be running down our leg. So these can be extremely long cells. However, in our brain, they don't have to be long. They're just going to be, potentially sending signals to the brain right next to a cell right next to them. Now, down at the bottom, now it gets rather confusing down here, so I'm going to focus on this neuron. But every neuron has these. They're axon terminals. And these axon terminals are going to be really important in sending the signal from neuron to neuron. So, on this neuron, it's really hard to see, but here they are. Here are the axon terminals. Now, the junction between the two, this connection between one neuron and the next, that would be a synapse. And the synapse is going to be the junction whereby the signal goes from the presynaptic, so this neuron would be presynaptic. The neuron at the bottom would be postsynaptic. And that should be pretty intuitive. It's pretty common sense. Here's our synapse, so here's the one before it, here's the one after it. Now, this diagram doesn't show Schwann cells. Schwann cells are very specialized cells that wrap themselves around the axon. We'll take a look later at what they do, what their job is. Now, there's a couple of really interesting characteristics of neurons. Number one, they are amitotic. They do not divide. And then the other one is they use a lot of energy. They consume a lot of ATP, and you're going to see why that is in just a second. And then the real important part to understand is the axon. How does this axon send a signal? So you can think about a situation. Let's go ahead and, maybe your little brother is playing with his Legos, or a little sister is playing with her Legos, and you're walking across the floor, and as you set your foot down, they left their Legos out, and you just land your foot right on that Lego. And you want to think of that dreadful pain, and that pain has to travel, that signal has to travel up your leg, and eventually that signal is going to get to the brain. But in the meantime, it's going to loop back, and by the time you are saying, ouch, you've pulled your leg back. And what we want to look at is how are we sending the signal. So to do that, we need to understand a couple of electrical terms. One is going to be voltage, and then the other one's going to be current. So let's establish some things. So first of all, we need to understand that if we were to take a look, forget about this era for right now, if we're going to take a look at the axon, the inside of the axon has some very, very unique combinations of ions. Voltage from an electrical term, voltage is the separation of charge. Or charges. Now, when we think about it from our batteries or an electrical plug, we're thinking of, okay, well, we've got those positive and negative charges, electrons, and as the electrons are separated out, then we can then have a current flow through them. Now it's a similar idea, but with our neurons and with invertebrates, they're going to be using ions. And so the important thing is going to be these ions. So the way this axon is going to be maintained, it's going to maintain a higher concentration of potassium ions on the inside, and then a higher amount of sodium on the outside. And we'll take a look at how that occurs. But it is really important that you understand, where that there are negatively charged ions as well. So we've got chloride ions, we've got phosphates, we've got all sorts of these main ions that are in there. And you could almost, although I am oversimplifying this, but you almost want to imagine that you've got 80 positive charges and 180 negative charges. Well, overwhelmingly, that's going to make the inside of the axon negative. Now, when we measure it, it's actually negative 70 millivolts. Now, that would be a voltage, although it's millivolts, but that's a measure of how separated the charges are in the axon. Now, how does the cell do this? How do the axons do this? Well, let's take a look at the next diagram. So notice we're keeping the colors here, but what we're going to do is we're going to look at a section of the membrane. So this is still the axon of the neuron. And then this is still going to be the outside and then the inside of the neuron. Now, and you can see the outside is in the blue, the inside is going to be in the beige. So what we have here are 3 different types of channels. Now, yours is not color-coded. You know by now if that helps you. And so we've got our sodium-potassium pump. And this is a nice review of active transport. The sodium-potassium pump is going to pump 3 sodium ions outside. So let's go ahead and write that down here. 3 sodiums out. And it's going to pump in to potassium. Now, that's an uneven amount to begin with, right? We're always pumping 3 positively charged ions outside. We're always pumping in only 2 positively charges inside. So that sets up an unevenness too. So you're constantly pumping and pumping and pumping these, and you do create a separation. It's not a huge amount, but it's just enough to set up a difference in charge, and that's a voltage. That's a separation. Now, you also have leak channels, and the leak channels are these openings and proteins that allow the ions to drift through. Because think about it, what we've just done is created, if this continues, we're going to have a high potassium concentration on the inside of the axon and a high concentration of sodium on the outside, which means that if they're allowed to, they're going to drift back over according to their concentration gradient. Potassium's going to go through to the outside, sodium's going to drift through to the inside. And what this does is create an interesting dynamic here. It's kind of constantly in flux, but what the cell always does, what the axon always does, is maintains this difference. And so notice you do have more potassium. This is a potassium leak channel. This is a potassium leak channel. This is a potassium leak channel. This is a potassium. And so here's our only sodium. So potassium is far more permeable. And we see that because there are more leak channels in the membrane. So more potassium ions can leak out. What in the end happens is this leakiness of the system means that it's not quite as negative on the inside as it could be. It could be negative 90 because of all the, but then the sodium drifts in and then some potassium drift out. And the balance of that makes the inside negative 70. So just to kind of sum up, we've got the sodium-potassium pump creating a high sodium, and I'm making that nice and big to kind of indicate that difference. A high sodium concentration on the outside, a smaller sodium concentration on the inside, just to change my colors and allow us to see things a little bit better. What we're also going to then have is a higher potassium concentration on the inside, and then a lower concentration of potassium on the outside. Now, because of those negatively charged ions, which don't forget they're there, because they are there, then we're going to have an overall negative charge on the inside, an overall positive charge on the outside. So that's due to the sodium potassium pump using a lot of ATP there to provide that difference. But it's also due to those leak channels that kind of create an interesting flux. You know, the ions are kind of drifting in and out, and then the pump is constantly maintaining that. This is so important that if you had anything blocking these leak channels or the sodium potassium pump, you're going to have issues. You're not going to be able to make your gradient. And there are some poisons and toxins, and we'll look at that next week, that can block and then therefore kill an animal or a person because they've done that. So we want to talk next about the action potential, but that is another video. This gets a little overwhelming to students, and I totally get it. Just go back and make sure your drawings are very nice and neat. We have a higher sodium on the outside, smaller potassium on the outside, higher potassium on the inside, smaller sodium on the inside. But overwhelmingly, because of the negatively charged ions on the inside, we've got a negative charge on the inside. That kind of swamps the positively charged potassium. Well, hope that helps. Please make note of any questions you have so that we can go over them on Monday. Have a lovely weekend.